Good day and welcome to our topic 2 of our basic level training lecture series on antimicrobial agent. And this topic 2 is still part of our module 1 on antimicrobial agent. I am Dr. Apalata, an associate professor of clinical microbiology and infectious diseases. So let me share my screen with you so that we can continue with our topic 2. So in this topic 2, we will mainly discuss about inhibitors of cell wall synthesis. So inhibitors of cell wall synthesis can be classified in 4 groups. We have the beta-lactams that are the widely used antimicrobial agent. Then we have the glycopeptide, we have the phosphomycin, and we have bacitracin. So the beta-lactams, we know them, they are composed of penicillins. That was the first antibiotic to be discovered. We have cephalosporins, we have the monobactams, and we have the carbapenems. And among the glycopeptide, we will mainly look at vancomycin and teicoplanin. And we will say some few words about phosphomycin and bacitracin. So we are saying that these are inhibitors of cell wall synthesis. And here we can give you an illustration of the bacterial structure, especially the bacterial cell wall. We can see here we have the peptidoglycan layer, and this is a peptidoglycan layer, and inside we have a cell membrane or a cytoplasmic membrane that is there, so those drugs that we are going to describe here, they actually act here on the peptidoglycan layer, especially, and we will see mainly the difference between the peptidoglycan layer of gram-positive versus the peptidoglycan layer of gram-negative bacteria. But it's important, since those antimicrobial agents inhibit or interfere with the synthesis of peptidoglycan layer, the peptidoglycan layer is a very complex structure, and its synthesis happens in different steps, okay? So we can have the synthesis of peptidoglycan monomers, then the formation of the linear sugar, then there is a cross-linking between peptide chain, and we have autolysin mediated breaking of peptide bridges. Okay, so peptidoglycan layer is a complex structure, as we can see here. And the beta-lactams are well known to prevent what we call transpeptidation, especially when there is a cross-linking between peptide chains. So it interferes with that mechanism, preventing it from happening and making the peptidoglycan layer more fragile and even the synthesis not to be complete or to take place, okay? So here, if we can see these two pictures, we have a gram-positive cell structure and we have gram-negative cell structure. It's just very important for us to understand the difference between the two, and that will make sense on how we actually use antimicrobial agent, okay? So the gram-positive cell structure, you can see it has a huge layer of peptidoglycan. You see here, the peptidoglycan layer is very thick, very thick, made of teichoic acid that are there. Okay, so it means that really an antimicrobial must be able to diffuse through before coming here into the inner membrane, where there is the target, like for the beta-lactams, because we are discussing beta-lactams, where you found penicillin binding protein. That is the target where actually your beta-lactam drugs will act. So they have to penetrate by diffusion until they will reach the PBP. So the difference between this structure and the gram-negative structure is that the gram-negative bacteria has a very thin peptidoglycan layer that we can see here. And it's also covered by something else that we call the outer membrane. Then the outer membrane has porins, and there are proteins here that are able to transport certain molecules, antibiotic molecule, into the cell before diffusing through the peptidoglycan layer to come and reach the PBP. So this structure on itself makes limitation if you are selecting an antibiotic, whether you are suspecting a gram-positive or a gram-negative to be the cause of an infection, because some drugs, for example, we will see that if they are not able to pass through the porine, or if they are not able to bind to specific proteins, they will not be able to penetrate. So we will try to see that. And this is the reason when we will talk about glycopeptide, this is one of the reasons why glycopeptide cannot be used against gram-negative bacteria, because it is a large molecule and it will not be able to pass through the porins that are there. So we say that in a gram-positive bacteria, you have a molecule. This molecule should diffuse through the peptidoglycan, this thick layer of peptidoglycan, and from there, it will then come and reach the penicillin binding protein that is the target for beta-lactam antibiotics. So you have the target here. So it will then reach that target, okay? But for the gram-negative, the antibiotic need to pass through the porins or be taken by a specific proteins that might play a role of a transport to take it through. Then from here, it can then be able to diffuse, and before it reaches the PBP, that is the penicillin binding proteins. So we have the mechanism of action. We say that it interacts directly with the PBP, and then its beta-lactam interact with the PBP, then they will be able to inhibit the transpeptidase activity, the transpeptidase activity that happen during the synthesis of peptidoglycan formation. So we have different classes of those drugs. We have mainly the penicillins. The penicillins, we have penicillin G and penicillin V that are natural penicillin. And we have the semi-synthetic like your ampicillin and amoxicillin. And we have those that are also semi-synthetic like your methicillin and cloxacillin that have a particular activity. So against staphylococcal infections. So the penicillin G and V have a narrow spectrum against gram-positive, and they can only act against very few gram-negative, but really their spectrum of activity is really mainly limited to gram-positive. So if you have to select penicillin G, beside things like treating treponemal infection like syphilis and so on, they are only restricted to treat gram-positive infection due to their narrow spectrum of activity. But when you have the semi-synthetic ampicillin or amoxicillin, although they have a narrow spectrum against gram-positive bacteria, but they have a high increased spectrum of activity against gram-positive, gram-negative, as compared to natural penicillin. So that's why ampicillin and amoxicillin can be used against both gram-positive and gram-negative, unlike your penicillin G. Then we have cloxacillin that has also a narrow spectrum against gram-positive bacteria only, including strains that produce penicillinase. So cloxacillin is really a good choice nowadays because of most of the staphylococcal infections are caused by Staphylococcus aureus that produce penicillinase. And that's why cloxacillin becomes the first choice against the staphylococcal infections due to high level of penicillinase that is produced by Staphylococcus aureus. Then beside the penicillin, we have the cephalosporins. We have cephalosporin C that are naturals. And we also have the first generation cephalosporin that are semi-synthetic drugs. Okay, so they have a narrow spectrum that is similar to penicillin, but with more increased gram-negative spectrum. So we will see that with the cephalosporin, as we move from first generation to second generation, third generation, fourth and fifth generation cephalosporin, their activity against gram-negative also increases. Okay, so like your second generation cephalosporin, they really have a narrow spectrum, but with increased gram-negative spectrum compared to the first generation. And the third and the fourth generation cephalosporin that are also semi-synthetic, they now have a broad spectrum of activity against both gram-positive and gram-negative, including even some beta-lactamase producers. Okay, so that makes the third generation cephalosporin especially a very good drugs. Whether it's a respiratory tract infection or abdominal infection or even against Neisseria gonorrhea infection, sexually transmitted infection. So it makes it a very good drug because of its broad spectrum of activity. Then we also have the fifth generation cephalosporin that has a broad spectrum against gram-positive, gram-negative, including among the gram-positive, it also has activity against methicillin-resistant Staphylococcus aureus. So it acts against MRSA. So we have different generations of cephalosporins on this slide. We can see the first generation, cefalexin, cefazolin, cefadroxil. The second generation, we have cefaclor, cefiroxim, cefoxitin, that is mainly used for testing in the lab, but cefiroxim is the most used in this generation. We have a third generation, we have ceftriaxone, ceftazidime, and cefixime, and cefotaxims, widely used member of the third generation cephalosporin. Then we have the fourth generation cephalosporin like your cefipim and cefpirom, and we have the fifth and the latest generation of cephalosporin. We like your ceftobiprole, ceftarolin, and ceftolozane. Those are members of the fifth generation cephalosporin. So as we indicated that the cephalosporin coverage, as we move from first generation to the fifth generation, we will see that the spectrum of activity also increases, although mainly the first generation have a better activity on gram-positive, but the activity against gram-positive is maintained throughout. But then the activity against gram-negative increases, And the most important thing is that with the fourth generation have anti-pseudomonal activity. So it can be used to treat gram-negative, including Pseudomonas infection caused by Pseudomonas aeruginosa, And also the 5th generation has a very good gram-positive cover, but it also have a cover against methicillin-resistant Staphylococcus aureus, and also it maintains the cover, like gram-negative cover, like the one we observe with the 4th generation cephalosporin. Okay, so we continue. Beside the cephalosporin, we also have the monobactams as part of the beta-lactams drugs. We have the monobactams that interact directly with PBP, and also they inhibit the transpeptidase activity. So we have, among the monobactam, we have aztreonam, that is a semi-synthetic drug, and it has a narrow spectrum against gram-negative bacteria, including some beta-lactamase producers. Then we have one of the broad spectrum, that is carbapenems. The carbapenems actually, like your imipenem, meropenem, doripenem, that are the semi-synthetic, they have a broad spectrum of the beta-lactams against gram-positive and gram-negative bacteria, including many of the beta-lactamase producers. So against them, we use carbapenems. So we can see here the carbapenems. If we look at their activities, we have 3 groups of carbapenems. We have group 1 carbapenems that is made of ertapenem, panipenem, and tebipenem. We have group 2 that is made of imipenem, meropenem, doripenem, and biapenem. And group 3, we have tomopenem and razipenem. So their activity, if we can see activity against, especially because we know that they have a broad spectrum of activity against gram-positive and gram-negative. And the group 1 carbapenem don't have activity against non-fermenters, gram-negative bacteria like your Pseudomonas aeruginosa and Acinetobacter baumannii. So don't use ertapenem, choose ertapenem if you suspect that the infection can be due to Pseudomonas aeruginosa or Acinetobacter baumannii. But the group 2, your imipenem, meropenem, doripenem have a good activity against the Pseudomonas, most of the gram-negative, including Pseudomonas and Acinetobacter baumannii. And the group 3 also have a good activity against those non-fermenter gram-negative bacteria. But activity against gram-positive, we say they have, but does that activity include MRSA? No for group 1 carbapenem, no for group 1 carbapenem, no also for group 2 carbapenems. Only group 3 carbapenems have activity against MRSA. So this is very, very important to know. Okay, so beside the beta-lactams, we also say that the inhibitors of cell wall synthesis include also the glycopeptide. And the glycopeptide inhibits the final cell wall stage, the stage of peptidoglycan synthesis by binding to the D-ala terminal. So when the cell wall synthesis, especially the peptidoglycan, is being synthesized, now when it reaches its final stage, that's where the glycopeptide acts. So we can understand that glycopeptide and beta-lactams are all inhibitors of cell wall synthesis, but they act at different stages. So the beta-lactams act a bit at the earlier stage, and the glycopeptide acts at the terminal stage by binding to the D-ala terminal. So they inhibit the actions of the glycosyl transferase and the transpeptidase by blocking pentaglycine from joining molecules. So there is no peptidoglycan layer growth that takes place. So now the problem with glycopeptide, like your vancomycin, is that they are a very large molecules. So when you give them, you must know that it will take time for them to act because they take time to diffuse through the peptidoglycan layer of gram-positive bacteria. Then after that, they will go and bind to their target. But in a gram-negative bacteria are intrinsically resistant to vancomycin, to the glycopeptide, simply because they are large molecules and they cannot, remember, the gram-negative has an outer membrane that has a porins. So you need a molecule that is able to pass through the porine of the gram-negative. And they are large molecules. They cannot diffuse through the porins before they diffuse through the peptidoglycan layer. So that's why glycopeptide cannot be used against gram-negative for the treatment of gram-negative bacteria. So we have 3 that have been clinically approved, 3 types of glycopeptide. I spoke of vancomycin, but we also have teicoplanin and televancin. Those are the approved glycopeptide. Here we have, in this study that tried to look at different reference strands of MRSA, methicillin resistant Staphylococcus aureus. They look at them. They look also VISA, that is vancomycin intermediate Staphylococcus aureus, and they look also Enterococcus faecalis. So those are different reference strands, but they try to compare the MIC of those 3 vancomycin. You know, we have the televancin, vancomycin, and teicoplanin. So you will see that against MRSA, if we look at the MRSA, the different strands of MRSA, where we can see that there was the lowest MIC, we can find here mainly teicoplanin against this, teicoplanin. So in general, but we found that vancomycin, despite its activity against MRSA, but the MIC was a bit higher as compared to teicoplanin and even televancin. Okay, but when we look at their action against the activity against vancomycin intermediate Staphylococcus aureus, the strand that was vancomycin intermediate, we will see that it's televancin that has the lowest MIC. So you can see those with a MIC of 8 that are vancomycin intermediates. Teicoplanin had even a highest MIC of 16, so you cannot choose it. So at least televancin will be a good choice for treating VISA. Okay, and the other strand, you can see that the activity was well determined for televancin, but the MIC was 1. Okay, so Enterococcus faecalis, we can also see vanco has a good activity, but the MIC is very high. You know, for faecalis, it's 0.75 here, teicoplanin 0.12. So teicoplanin was a better choice here against faecalis, followed by televancin, then vanco. Fishium, we can see 0.12 here, 0.12. Televancin with the same result like teicoplanin, and vanco slightly high. But fishium, there is another very resistant one that vancomycin cannot even be used against. But televancin and teicoplanin had a very lowest MIC according to this study. Okay, so those were inhibitors of cell wall synthesis, and we try to focus more on beta-lactams and glycopeptide as they are the commonly used antimicrobial agent. Then we will try to look at in some of 2 knowledge tests to try to illustrate our understanding. So the test one, okay. All the antibiotics listed below refer to the beta-lactams except one, except. So pause your video for 30 seconds, try to think which one is not a beta-lactam. Okay, so we said, if you remember what we said, okay, we said that beta-lactams include the penicillins. So penicillin is part of beta-lactams. Then we have the cephalosporins, we have the monobactams, we have the carbapenems. So the answer is D. So except cycloserine. So cycloserine is not a beta-lactam. Okay, so the rest, monobactams, cephalosporin, penicillin, carbapenems, are all beta-lactam. Okay, now the second test is that each statement formulated below is correct regarding the activity of vancomycin, except. So try to read these different statements and try to find out which one is not correct in relation to the activity of vancomycin. In relation to the activity of vancomycin, which statement is not correct. Okay, you have 30 seconds, pause your video, and come back. Well, let's go through this statement one by one, okay? The first one, vancomycin is active against gram-positive bacteria. That is correct, okay? Vancomycin is active against gram-positive bacteria. The second one, vancomycin is inactive against gram-negative bacteria. This is also true. Remember, we said vancomycin is a large molecule and gram-negative are intrinsically resistant to vancomycin simply because gram-negative have what we call an outer membrane, and that outer membrane covers their peptidoglycan layer. And because the outer membrane have porins where the molecules, the antibiotic should pass through, vancomycin has a very high molecular weight. It cannot pass through those porins. So vancomycin is inactive against gram-negative. So this statement is also true. Vancomycin isn't inactive against the MRSA. So this is also true. We say vancomycin is used to treat MRSA, methicillin-resistant Staphylococcus aureus. So it isn't inactive. It is active against the MRSA. Vancomycin is active against streptococcus. This is also true. It can be used to treat a streptococcal infection, as streptococcus is a gram-positive bacteria. Vancomycin isn't active against Enterococcus faecalis. So this statement is not true. It's not correct because vancomycin, we have seen that it has activity against Enterococcus faecalis. But we have seen high MIC of vancomycin, mainly against Enterococcus faecium, especially if we compare that with other glycopeptide drugs, like the other two that we have shown here, like your televancin and tacoplan. Okay, so the answer here is E. So the statement E is not correct. Okay, so follow us when we will discuss our topic 3, that is inhibitors of cell membrane functions. And we are going to discuss the topic 3, and we continue with our module 1 regarding antimicrobial agents. Thank you very much.